So my name is Charlie Loewen. I'm an assistant professor in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. And we are out here today at one of our field sites for an INRC project looking at the co-benefits of nutrient reduction wetlands and whether or not they may have any sort of unintended consequences that we should be aware of as we are continuing to put these on the landscape. So what are some of the unintended consequences? Well, obviously, Iowa has a tremendous amount of agriculture, and so one of the consequences of that is following rain events and snow melts. A lot of the runoff and tile drainage that comes from these fields often has very, very elevated levels of nutrients. And another consequence of all this agriculture is that we drained many of the naturally occurring wetlands across the state. And this creates a problem because wetlands are actually very, very efficient and good at reducing nitrogen loads and phosphorus loads sometimes before they reach downstream. And so some of the unintended consequences may be that, for one thing, they are much more efficient at reducing nitrogen loads than phosphorus. So we're fundamentally changing the ratios of some really key nutrients. And this is important because different organisms have different nutritional requirements. And certain organisms, such as cyanobacteria, which are a type of phytoplankton, or that live in the water column or attached to substrates, can sometimes produce their own nitrogen from the atmosphere. And so if we're alleviating some of the nitrogen pollution without having similar drops in phosphorus levels, we could be creating situations where cyanobacteria, some of which can produce harmful toxins, may be able to have a competitive advantage over other organisms, and it could possibly lead to more cyanobacteria downstream. It also is possible that just by reducing nitrogen levels overall and phosphorus to the extent that they do, we may have a drop in that, but we don't know until we look. So that's one of our goals, is just to see whether that is something we need to be concerned about going forward. So the Iowa Nutrient Research Center funded this project because restoring wetlands are one of our leading edge of field practices for reducing nutrient loads from agricultural fields. And so we wanted to go and look at, well, we're putting these things in the landscape. Are there other co-benefits for other biological values that we can try to maximize or optimize as we go forward and continue to create these wetlands or restore them on the landscape? So big part of one of our main objectives is to go out here and look at some of the lower trophic levels. So these are things like that fuel the broader food web, and looking at the health of those communities as an indicator of the quality of the habitat and the ability for these wetlands to provide useful habitat to water birds and other animals that we maybe want to provide more habitat to on these agricultural landscapes. One of the really valuable parts of field research programs, or just research in general, is having the opportunity to train students. So for this project, I was lucky enough to work with 3 graduate students who all are able to look at some of the biodiversity in these wetlands at different levels to focus on different communities. So with that, I'm going to turn that over to some of my graduate students to show you how they are sampling these communities to try to answer some of these questions we have about the functioning and biodiversity values of restored wetlands.
My name is Daniel Edwards. I'm a PhD student in the Loewen lab. I am originally from Indiana, but I'm here in Iowa doing my PhD. I specifically study aquatic macroinvertebrates, which is this kind of big group of aquatic organisms that consist of a lot of different groups of insects, as well as some crustacea, some snails, and the likes. And this is a really interesting group to kind of work with because they've all kind of independently moved into these landscapes. And so they all kind of have these very different strategies to survive within these landscapes. And so because of this, different groups are more or less sensitive to types of environmental stress or change, different types of respiratory systems. If you imagine, a lot of different of these insects have gone from this terrestrial environment where they're breathing air to then colonizing the aquatic landscape where they're then breathing through gills or siphons. All these kind of different strategies allow different groups to being more or less sensitive to different types of stress in the environment. So these aquatic macroinvertebrates are really important for a lot of different functions. They do serve as this really interesting kind of aquatic terrestrial linkage. A lot of these organisms do not spend their entire life cycles in the aquatic environment, and so are actually exporting nutrients into the terrestrial landscape when they get eaten by birds or other organisms. They're also kind of this really nice kind of intermediate step between the aquatic like planktons and the invertebrates, and then they get eaten by fish or birds and other things. So they kind of are this really nice kind of middle level of the environment. My role in the research is to do the research. I'm out sampling the bugs. In these wetlands, I use several different strategies to do so. I sample macroinvertebrates upstream and downstream of the wetlands. And then within the wetlands, I use a novel strategy using this big trash can that I put into the wetland And then within this, one of the interesting things that I'm looking at is this linkage between the plant communities in these wetlands and the aquatic macroinvertebrates. So how the different plant life and the different growth forms of those plants can kind of affect one another. And so I take this big trash can and I put it into the wetland, and this is my sampling unit. All the plants get taken out of this and then get rinsed off from the bugs. And then all the bugs I then take out of this with the net. We sampled once a month for 3 months. These communities change a lot over the year. So one of the things that we're thinking about doing in the future is looking at sampling year round because these communities do change a lot as different organisms emerge out into their terrestrial life stage and then subsequently later colonize the wetlands again. I've always liked insects. The aquatic ones are a little bit more interesting because they're pretty generally understudied. They're super diverse. The aquatic beetles are probably my favorite. There's around 14,000 species worldwide, which is pretty crazy. And all of them kind of fill different niches and have really crazy adaptations to survive in the aquatic landscape. That's just really fun and really cool.
Hi, my name is Kaitlyn Kelly. I'm a member of Dr. Charlie Loewen's lab. I'm a master's student. I'm a first year. I'm initially from Southwest Iowa, and I was interested in the project going on up here at Iowa State, and so I became a member of the lab. My primary role in the lab is to look at the zooplankton communities, and so as our project looks over a bunch of different communities within these wetlands, I am specifically looking at zooplankton, which are microscopic invertebrates, and they are primary consumers of phytoplankton and algae. And so therefore it's important to look at them and what their communities are doing in terms of these wetlands and in terms of food webs, because they are important roles in the food webs and they provide food for the macroinvertebrates and the larger vertebrates such as fish and other things living in the wetlands. Zooplankton provide food for both macroinvertebrates like different bugs, and then they also provide food for other vertebrates such as birds and amphibians and other reptiles that might be living in the wetlands. So I have a couple different sampling techniques, but I have one that's more interesting that I'll show you today. And so that will be using a vertical Van Dorn, which is basically this clear tubing, and it has plungers on both ends. So you'll open both ends of the plungers. You'll send a messenger weight down and it will hit a button where it will close those plungers. And so traditionally the Van Dorn will be placed in the water column at this time. And then once you're at a specific depth, you'll send the messenger down. It'll close the plungers and it will trap the water where the Van Dorn is sitting at that moment. So I've always been interested in anything aquatic, and so I was working a position full-time for the Crumpton Lab before I became a master's student. And so I was already quantifying zooplankton for their ambient lake monitoring project, and so I was really interested in also looking at zooplankton communities within wetlands rather than lakes, because lakes have been very traditionally looked at in terms of zooplankton communities, so it was something new to learn about.
Hi, my name is Grace Jackson. I'm originally from Dayton, Ohio, and I moved here 2 years ago to obtain my master's degree in the Loewen Lab. My role in the lab is studying the base of the aquatic food web, which is phytoplankton and periphyton. Now, these two groups are sort of like plant-like organisms, but they include a wide range of life, including bacteria and protozoa. And they have the ability to photosynthesize. So they take energy from the sun and convert it so that other organisms, such as the zooplankton that Kaitlyn talked to you about, and eventually up the food web into the macroinvertebrates and the amphibians, can obtain that energy too. Phytoplankton, phyto meaning obtaining energy from the sun, and then plankton meaning they're floating freely in the water. So that is one group that I study. That's the group that forms the algae blooms that you may have seen around the state as a result of the nutrients coming in, because phytoplankton use nitrogen and phosphorus as well. And then periphyton are just a different life strategy that these plant-like organisms can use, where they attach to a substrate, such as a rock, and they grow almost like a microscopic forest structure. So I'm studying those two groups. I measure phytoplankton similarly to how Kaitlyn samples her zooplankton, basically by taking a surface grab of water, and then we use a special instrument called a flow cam, which is basically a fancy camera and microscope. You put your water sample in at the top, it goes through this microfluidic tube, and then there's an objective that's positioned right next to the microfluidic tube, and it captures tons of images of these microscopic plankton coming through. And then I can take those images and actually train the computer to recognize certain shapes, and it will help me sort these different groups of phytoplankton, and then I identify them just based off of traditional methods from that point. One of the objectives of this project is to understand how this wetland is affecting the biological communities up and downstream. We looked at periphyton, which again is the attached algae, using these special samplers. It's basically a flotation device that carries 16 microscope slides. We tie this device to a cinder block with some string, and we deploy it, and it floats on the water surface or just below the water surface for about a month, and we come back, and there's a bunch of periphyton that have grown on these slides. And then we can take those slides back to the lab for analysis, either using the flow cam, or we've also frozen them for pigment analysis. And then we can do pigment analysis in the field using a handheld spectropluometer. And basically what that is it has a light source and a lens, and you put it to the microscope slide that has a bunch of periphyton that's grown on it, and it flashes, and then it gets a reading back, and whatever the reflectance back from the periphyton is, every kind of algae has its own special amount and types of pigments. And there's a computer in this handheld spectropluometer that recognizes those particular ratios and types of pigments in order to identify three main groups of the periphyton, which is cyanobacteria, diatoms, and green algae. One of the big questions when we started studying these wetlands is there more cyanobacterial growth before or after the wetland, or is there really no difference? And the reason why we were interested in that is because certain species of cyanobacteria are known to be what is known as nuisance species, because they can produce toxins that affect our water quality and our public health.
So that largely concludes the outdoor portion of this, where we've kind of demonstrated how we are sampling some of these wetlands. Again, our objectives here are to understand the biodiversity within these restored wetlands so that we can, hopefully as we continue developing them on the landscape, perhaps use some of our learnings to build them in ways that may further optimize some of those biological values, as well as avoiding any potential problems if we find them. Very exciting as we continue to build these on the landscape to help improve our downstream water quality, and again, hopefully try to associate increasingly additional values for biodiversity, things like water birds, to help them on these very agricultural landscapes.